We're moving on to step 2 and step 3, the breakdown of pyruvate and the Krebs cycle. We've already covered what cellular respiration is. We've discussed aerobic versus anaerobic reactions, and that cellular respiration is an aerobic process requiring oxygen. We discussed how glucose is a simple sugar. It can be broken down systematically. The first part of this process occurred in the cytosol, and we're going to start moving into the mitochondrion in a eukaryotic cell. So we're going to begin with how to break down pyruvate. We're going to end with the Krebs cycle. We'll produce a whole bunch of energy intermediaries. And then from there, we'll move forward into the final step. Let's start with the breakdown of pyruvate. All right, step 2. That's all step 1. Everybody, any questions on glycolysis? All right, don't worry. We've only got 4 steps to get through. And then we got 2 more steps to get through fermentation. And, we're going to talk about, okay, so there's a little bit more. There's more to it. But we're almost there for this one. Little bites. Nope, all of it. Breakdown of pyruvate. You're going to take these 2 pyruvate. You're going to break them down. Now, this can happen in the mitochondrion. It's important to note that, yes, you do break down pyruvate in the prokaryotic cells with no mitochondria, but not in this manner for this process. It can happen, but not for the process I'm about to show you. What we're going to do is we're going to take that pyruvate. How many carbon are in pyruvate? 3. And we're going to hack one of them off. All right, so we're breaking a bond. What happens when you break a bond? Energy is released. Energy is going to be released in the form of 2 NADH. 2 pyruvate, 2 NADH. You've hacked off that carbon, though, in the form of 2 carbon dioxide. Now remember what our original equation was? C6H12O6 plus 6O2 yields 6 carbon dioxide and 6 water. Here's 2 of those carbon dioxide. So far, so good? Pardon? To rebuild the glucose? No, because that's not happening in this organism. So we created NADH. We've pulled in those high energy electrons. So if we've moved electrons from pyruvate to NADH, what have we done? Well, it's stored energy. Let me put it a different way. We've oxidized the pyruvate. Thanks. That remaining piece is an acetyl group. So you get an acetyl over here, 2 acetyl groups. And those acetyl groups get attached to a special enzyme called coenzyme A. So you attach these 2 acetyls to coenzyme A, leading to 2 acetyl-CoA. Where have we seen coenzyme A? Holy cow, 2 of you know it right off the bat. Where have we seen it? That's right, it activates phospholipid tails. In all of the time I've been teaching this, nobody has been able to come right out. Usually this is where I take a break and get water. Nobody else has done that, so very well done. And there were 3 of you, so that's mind-blowing. So now we've got 2 pyruvate, which is 3 carbon. You cut off a carbon from them, so how many carbon are left? 3 minus 1 is... Because, yes, there are 2 pyruvate, and you cut off 2 carbon dioxide, so you end up with a 2 carbon molecule there. All right, check this out. Pyruvate has 3 carbon in it. You cut off carbon dioxide from the pyruvate. Carbon dioxide has 1 carbon in it. So now you go from 3 minus 1 is 2.
So why is there 2?
Because there are 2 pyruvates. So they each they release 2 carbon dioxide to produce 2 acetyl coenzyme A.
So there's 4 carbon total in that step? Or...
There are 4 carbon total, but 2 carbon in that molecule.
Okay.
Yes.
Can you circle the 2 in the pyruvate so I know they're together too? I didn't
Like that?
Yeah.
Want me to do that with this too?
Yes, please.
No problem. So far, so good? Questions? All right. Of course, you guys know now, this helped attach the phospholipids. It was apparently on the next slide the whole time. So the whole point of this step is you've released that waste product, carbon dioxide, you've created 2 NADH, and you created 2 acetyl coenzyme A. Step 3, the citric acid cycle, otherwise known as the Krebs cycle. So at this point, this occurs inside the mitochondrion. Remember, the mitochondria is a double membrane-bound organelle. What are the other 2 double membrane-bound organelles? Chloroplast and the mitochondrion and the nucleus. All right, now the mitochondrion has this double membrane that's got all these twists and turns. What do those twists and turns do? They increase the surface area because it's at each of these twists and turns that some of the reactions we're going to go into happen. This Krebs cycle happens inside the mitochondria, in the middle. So, history lesson, yes?
But the last time we did 6, we said we'll do some of it before we started the, I guess, what was supposed to be this. The H plus ions were outside of the,
We're going to get to that toward the end. They're going to go right, you're going to form your gradient right inside of here.
they come in with the ADP to produce
Yep.
ATP through some channels.
Yep. Yeah, that's the 4th step. We're almost there. We're almost there. You're doing a great job. You're applying it exactly right. That is coming up. We just need to get the electrons from this step to power pushing this hydrogen. So this is how we get the energy to do that. But I want to give you guys a history lesson on Hans Krebs. Hans Krebs discovered the Krebs cycle in the 1930s. You can imagine it was a tumultuous time. What was happening during the 1930s? Alcohol. Wow, that's worldwide.
Oh.
Worldwide. Depression. Anything else kind of important? World War something. 2. There we go. Okay, good. Yeah, that was kind of a big deal. But I appreciate that your mind went right to alcohol and money. Welcome to America. So Kreb discovered this in the 1930s. He did it by looking at pigeons. Now, you got to understand, Kreb himself was not Jewish. His wife was Jewish. And they lived in, I want to say they originally lived in France. He sent his wife away and gave her a cover name to hide her. He ended up moving over to England where he did his research. So she's hidden away. He's working on this stuff. And kind of the Nazis were hunting his family. He was not safe because, as you can imagine, London during the World War II was not a safe place to be. He spent his time studying pigeons. Oh, pigeons, you guys know what pigeons are, right? You've seen them? Pigeons are pretty much rats with wings. They are horrible, disgusting birds. And if there's one thing I've learned from watching movies, is if there's a crazy old lady feeding birds, be nice to her because she's either magical or she will save you at some point. It happened in Mary Poppins, happened in Home Alone. I mean, wherever you see crazy bird lady, she's not actually crazy. So that's my advice to you. Be nice to crazy old bird ladies. Important biology lesson there. In Mary Poppins, that's the one at the top. They were singing, and so I get to sing on YouTube. It's like, feed the birds, tuppence a bag. Do you remember that? Have you ever seen Mary Poppins?
No, that's what I'm trying to remember. I don't remember that.
That is totally Mary Poppins. Thanks. Appreciate it.
No, I believe
Okay.
you. I just can't remember.
Feed the birds. Someone always feed the birds.
Yeah, see?
In my house for some reason, they didn't get it.
Now, here's what you do to pigeons. You cut their wings. So now that we're talking about, be nice to birds. Don't be nice to birds. Check out what their wings do, because their wings have muscles. What Krebs was doing was he extracted muscle from the wings of pigeons to see how those muscles got energy. And he went through systematically and applied inhibitors to slow down those reactions. Now, what's an inhibitor? An enzyme inhibitor. It's okay. It's something. Yeah, competitive and non-competitive inhibitors, what do they do? But how? Right, so these inhibitors would either bind to the active site where the substrate would normally bind, or they bind to the allosteric site, changing the shape of the active site. So what Krebs did was he specifically blocked the enzymes in the Krebs cycle to see where you have a buildup of molecules, of chemicals. So he saw that certain chemicals worked in the cycle. You went from one to the next, to the next, to the next, and around and around and around. Each of those enzymes had to work, or else the cycle would fail. So he went on to win the Nobel Prize. The Nobel Peace Prize, you get a gold award. Oh, like all scientists want to get this. Lots of attention, and $1 million. Now, this is one thing I don't get about scientists who win the Nobel Prize, is inevitably, most of them win the Nobel Prize, $1 million, and they turn around and donate it to their college. For the record, that is not something I would do. I would win the Nobel Prize and then take the money and wave. But apparently other scientists who do win this are much nicer than I am. Also, I think the Nobel Prize looks sort of like those candies, those really cheap candies you get at Easter. You know what I'm talking about? Like the coins that nobody actually really likes. Same pigeons, of course. They're totally the same pigeons. They went on to... No, they're not the same pigeons.
We need to know that pigeon history thing.
Do you need to know pigeon history?
Know what the Kreb cycle.
No, you don't need to understand pigeon history. But I appreciate you bearing with me while I taught it. Can I add a question about pigeon history? Do you guys really want to know about pigeon history?
Oh yeah, that is true.
I'll make a note to myself to think about putting something about pigeons on the exam.
Explain about Kreb cycle, like what you do with pigeons.
Don't do it. Don't do it. An exam on Mary. How Mary Poppins relates to biology? I could do that.
Oh, he can. He probably can.
Journal of Science. It's not a bad idea. We'll talk about that later. All right, so here's basically how the Krebs cycle works. You're going to take this what is that thing? I don't know. This acetyl coenzyme A. And that acetyl coenzyme A is going to get systematically broken down in a cycle. A little that way. First thing that happens is you're going to hack off a carbon dioxide. You remove CO2. You've broken a bond now. All right, if you break a bond, what else is released? Energy. In this case, it's the form of NADH. So far, so good? So we've broken down that acetyl coenzyme A and cut off carbon dioxide. This acetyl coenzyme A, the first step is that acetyl coenzyme A binds to a 4-carbon molecule before it breaks off the CO2. Yes?
Why do all these things proceed?
Like this one? So you broke one glucose into 2-pyruvate. Does that make sense? So that's where that 2 comes from. 2-pyruvate get broken down. Each gets broken down into a single acetyl coenzyme A. But you have 2-pyruvate, so it's 2 acetyl coenzyme A. Right. So now you add the acetyl coenzyme A to a 4-carbon molecule.
And each acetyl coenzyme
Gets its own. So it's like 2 4-carbon molecules.
Does it do like one acetyl coenzyme A at a time?
Yes. It cranks through once and then twice.
Okay, so the circle and the numbers that you're writing, they only apply to one.
They are for one acetyl coenzyme A. Yes.
Okay.
Yes, so this is, you're taking the one acetyl coenzyme A and cranking it through the cycle one time. Then you're going to take the second one and crank it through a second time.
So it happens separately and not simultaneously?
It depends on how many enzymes are present. All of this stuff could be occurring simultaneously.
Just not on the same enzyme.
Yes, not on the same enzyme. So you could view it either way. I tend to view it as a sequential process because it's easier to keep track of in my head. But in the cell, they don't really care what works in my head. It just all happens. So carbon dioxide, NADH. Then we're going to go and we're going to cut off another carbon dioxide. You've broken a bond, so what else is released? Energy is released, NADH. Then you're going to hack off a molecule called guanosine triphosphate. We'll get into that in a second, GTP. It's an energy molecule. Then we're going to hack off another functional group, and that's going to release FADH2, an energy transport molecule. And then we're going to cut one more off, and we get NADH. All right, here's the big takeaway. Big takeaway. Yes, what we're doing here is we're producing a lot of energy intermediaries. Each of these has very, very highly charged electrons. These electrons have energy. They can do work. Does that make sense? Electrons have energy and can do some work. This is the Krebs cycle. I'm going to go into a little bit more. Quick note on guanosine triphosphate, GTP. GTP is just like ATP. It's got about the same amount of energy. The difference is it's useful for building proteins, whereas ATP is useful for building other macromolecules. The difference between ATP and GTP is the difference between a Canadian dollar and an American dollar. A Canadian dollar has about the same value as an American dollar, but you can't spend it in the same stores. Oh, up here, these are the details of the citric acid cycle, where you'd have to know each and every oxaloacetic acid becomes acontic acid, and isocitric acid to oxalosuccinic acid, and so on. This is what a lot of students have to learn. I'm not asking you to learn that. I'm asking you to understand the basic process. So yes, I know you think I'm asking a lot out of you to understand the process of aerobic cellular respiration, but I'm not being as bad as I could be.
Like later on, will we need to know that?
Not for 102, not for 101 with me. If you were to take cell biology, it's a 300 level biology class. That's, in my opinion, when you would need to know that. That is not offered here. Net results from one crank through of the citric acid cycle: carbon dioxide, 2 carbon dioxide, 1 GTP, 3 NADH, and 1 FADH2 per acetyl coenzyme A. But remember, it cranks through twice. The good news is we've got through 3 of the 4 steps. We've underwent glycolysis, so we split the glucose molecule into 2 molecules of pyruvate. Then during this mini lecture, we discussed how pyruvate itself is broken down by an enzyme, pyruvate decarboxylase. Pyruvate decarboxylase, it's a mouthful, will remove carbon dioxide. Decarboxylase removes carbon dioxide from pyruvate, and that means it forms, instead of one 6-carbon molecule, it'll form 2 2-carbon molecules. These 2-carbon molecules are known as acetaldehyde. That 2-carbon molecule will then bind with coenzyme A to produce acetyl coenzyme A. This is the way it's spelled. And in that process, a molecule of NADH is produced. So that's the breakdown of pyruvate. From there, the acetyl coenzyme A enters into the Krebs cycle, otherwise known as the citric acid cycle. And this is housed inside the mitochondrion. Acetyl coenzyme A gets attached to this new 4-carbon molecule. And I'm not going to ask you to remember what that 4-carbon molecule is called, but it gets attached to a 4-carbon molecule to make a 6-carbon molecule. And that 6-carbon molecule is systematically broken apart. You break a bond, you break a bond, you break a bond. And anytime you break a bond, you release energy. And that energy gets stored in energy intermediaries. In this case, the energy intermediaries are NADH, GTP, and FADH2. And also carbon dioxide is released in this process as well. 3 steps down, 1 more to go. These content review questions are here to focus your studies on from this mini lecture. Going forward, in the next mini lecture, we're going to discuss in depth oxidative phosphorylation and chemiosmosis, how the adenosine triphosphate is actually built. So that's coming up in the next mini lecture.